In 1991, the World Health Organization acknowledged that the predominant source of human exposure to mercury is from your fillings. That should be of concern to anyone. So all mercury-silver fillings leak substantial amounts of mercury constantly. The amount increases with any kind of stimulation, and as a result, mercury from fillings produces the majority of human exposure to mercury. The International Academy of Oral Medicine and Toxicology is extremely concerned about the anecdotal claims of safety by manufacturers and dental trade associations that are at variance with the published, peer-reviewed scientific evidence to the contrary. The precautionary principle requires action once the possibility of harm exists. It does not require proof beyond a shadow of a doubt that in the case of heavy metal and xenobiotic exposure is both nearly impossible and unnecessary, in our opinion. What you're seeing is mercury vapor coming off a 25-year-old silver amalgam filling in an extracted tooth. The background is a phosphorescent screen. The mercury vapor absorbs the fluorescent light, and you can see it as a shadow on the screen. This is mercury coming off a filling that was dipped in water that's the same temperature as the human body. This is a filling that was rubbed with a pencil eraser for just a few seconds. Like going to the hygienist and having her clean your teeth. These are not small amounts of mercury. If you can see it, it's more than 1,000 times higher than the Environmental Protection Agency will allow for the air that we breathe. What about the last time you went to the dentist and they drilled on your tooth? Here is the mercury vapor every time you raise the temperature to 110 degrees with hot coffee or warm water, or even chewed on it. Mercury comes off fillings every time you stimulate them, and that stimulation causes the mercury to continue to leak out of the fillings for an hour and a half at a minimum. Some people grind their teeth. Some people chew gum. The dentist might send an old gold crown to the dental lab to be welded. How about the dental personnel? They're not being given informed consent. Back in 1985, the International Academy of Oral Medicine and Toxicology set out to determine the amount of mercury that was coming off fillings. And here's the graph, showing substantial quantities of mercury were measured coming off fillings. And then we estimated the total dose. Then we began animal experiments and put radioactive fillings in sheep. Mercury accumulated in the jaw, stomach, liver, and kidney of the sheep in just 30 days. Substantial quantities of mercury spread from the fillings to every organ in that sheep's body. Then we measured that the sheep's kidneys dropped in their ability by 60% to clear inulin, an indication of kidney malfunction. Whole body imaging of monkeys found exactly the same thing. Proponents of amalgam fillings claim that sheep chew too much. Well, what's the problem with monkeys? They had mercury in their jaw, kidneys, liver, intestine, and heart. And further research found dystrophic bacteria that were antibiotic resistant cropped up in the intestines within 2 weeks of receiving these mercury-leaking fillings. Further studies have found damage to the ADP ribosylation of brain neuron proteins. In response to the controversy, and at the request of the Federation of Experimental Scientists and Biologists, Drs. Fritz Lorscheider and Murray Vimy wrote an editorial, the first ever in FASEB, that point by point refuted the claims of the amalgam proponents. That should be of concern to anyone wanting to have healthy children, because mercury is highly damaging to fetuses. Experiments in sheep showed that mercury from the sheep's fillings transferred immediately to the placenta, to the unborn fetus, and to every conceivable portion of the fetus's body. It even increased in the lamb higher after birth from mercury in the mother's milk. There's no such thing as a safe mercury filling. All mercury fillings leak mercury. The combined effect of mercury, cadmium, and lead is just now being investigated, but it's not 1-in-1. It's synergistic, and 1 and 1 may make 100 or even 1,000. Why is that of concern? Over and over again, we've heard that children are exposed to lead from our environment. Mercury in lead is many times more toxic than just mercury alone. These black, corroded, pitted mercury fillings are used where you must drill away 1/3 of the tooth in order to fix a pinhead-sized cavity. Even if you love mercury, it's the wrong thing to do to the children. It leads to broken, diseased, root canal extracted teeth throughout the rest of the life. It's a blunder that costs the child all through their life. Millions and millions of dollars are spent annually fixing teeth again and again. And dentists don't follow the manufacturer's recommendations. They pack mercury in children around gold crowns, underneath bridges. They stuff it around the gum line in contact with tissues. There's mercury spreading from this gold crown to every tissue in that patient's body. Even if you like mercury fillings, putting that kind of filling in the tooth is simply the wrong thing to do. Harold Lowe, the former director of the National Institute of Dental Research, back in 1993, wrote, "The first filling is a critical step in the life of a tooth. Using amalgam for the first filling requires removing a lot of tooth substance, not only diseased tooth substance, but healthy tooth substance as well. So in making the undercuts, you sacrifice a lot, and this results in a weakened tooth. The next thing you know, the tooth breaks off, and you need a crown. Then you need to repair the crown. And so it continues to the stage where there's no more to repair, and you pull the tooth. With the first filling, you should do something that can either restore the tooth or retain more healthy tooth substance. Use new materials, composites, or materials that can bond to the surface without undercuts. You can do this with little removal of the tooth substance so that the core of the tooth is still there. I would add that the cost of all that dental repair over and over again makes the cost of mercury fillings enormous, even if you don't consider the neurological impairment and the brain damage that they surely cause in dental personnel, and the infertility and the heartbreak that they've caused to so many families. It is the opinion of this Academy that responsible government agencies should prohibit the use of these fillings until such time as their manufacturers produce the alleged evidence of safety.
I've seen the smoking teeth video, and I show this to all of my patients who discuss to me mercury toxicity. They say, well, how could this mercury filling be causing me problems? I've had them in for 30 years. The video ends that discussion and brings the seriousness to the patient of what a problem this is.
Some people might question that the smoke we see coming off the amalgam filling might not actually be mercury. The fact that it is mercury can easily be demonstrated with a Jerome mercury vapor analyzer. The tooth is first cooled and then analyzed with the mercury sniffer. The Jerome shows that there are no emissions coming off a cooled dental amalgam. Then we stimulate the surface with a small explorer, scraping gently, and the Jerome takes another 10-second sample. California maximum contaminant level for mercury at the hazardous waste dump is 100. This filling is well over that level in just seconds of stimulation, thus demonstrating beyond doubt that amalgams leak mercury anytime they are stimulated.
In a class that I teach at the University of Kentucky called Mercury Science and Politics, I have the students do the same experiment as you see in that smoking tooth video. We use a fluorescent screen, an amalgam filling made outside the mouth, and a UV lamp. And we turn off the lights in the classroom. I have the students do this with their own hands, and they hold the amalgam in front of the fluorescent screen, and they see this vapor coming off, and we tell them that's mercury. And the way we establish that it's mercury is that we use a mercury vapor sniffer, and we take it up to the amalgam tooth and take a sample, and it shows that that is indeed mercury, that is the vapor coming off. We then have the students take a toothbrush, a store-bought toothbrush, and they brush that amalgam filling for 30 seconds as if they were brushing their teeth, and they pull it away and they look, and indeed the amount of mercury vapor coming off has increased dramatically. Again, we use the mercury vapor sniffer. We take an analysis of that vapor, and what it shows is that the mercury vapor coming off that tooth has gone up about 10-fold.
It continues to amaze me how blind medical and dental profession has been about the seriousness of mercury toxicity. For well over 100 years, the severity of mercury and the toxicity of it has been well recognized. But even to this day, the dental profession continues to put 100 tons of mercury fillings in the mouths of Americans every year.
I had a patient in her 50s who was in perfectly good health when she read information about amalgam fillings being potentially dangerous, and as a result went to her family dentist requesting to have her fillings replaced with non-toxic material. And in the course of 2 days, she had 18 fillings removed without any type of protection to prevent mercury vapor from entering her system. Within 2 weeks, she began developing irreversible neurologic damage that was ultimately diagnosed as Parkinson's disease, and now she is permanently disabled.
I think one of the major things that I'm concerned with is I go around telling people we should get rid of amalgam fillings. They shouldn't be used. This may cause some people to rush out and get their amalgams removed by someone who won't take care of removing them safely. Research has shown that if you do not take safety precautions, the mercury level in your blood can go up 3 or 4-fold higher at least. Because when you put the drill on the amalgam filling, you heat it up and vapors start coming out, and the patient and the dentist start breathing those vapors. If you use a safe procedure, such as putting an oxygen mask on the dentist and on the patient, and using certain techniques that have been developed by the IAOMT dentist, you can decrease the amount of mercury exposure to the patient and to the dentist. And this is critical because anyone that's not feeling well or cannot excrete the mercury is going to get a large dose of mercury on having their amalgams removed, unless they use the techniques that prevent them from breathing the mercury vapor that's being generated during the removal.
Recently, a 52-year-old physician in good health decided to have his mercury fillings replaced. He immediately became ill within 2 weeks and would have died had he not had a heart transplant. He had developed idiopathic dilated cardiomyopathy. Research has linked this often fatal heart problem to mercury. IDCM heart has 22,000 times more mercury in it than a heart that's sick for cardiovascular reasons. You need to be protected when mercury fillings are removed. Everyone, including the European Union, the American Dental Association, the amalgam manufacturers, and the International Academy of Oral Medicine and Toxicology agree that removing old mercury silver fillings can be dangerous because it exposes everyone present to enormous amounts of mercury. Fortunately, there are steps that any responsible dentist can take which will greatly reduce the risk and minimize everyone's exposure to this toxic metal. First, the dentist needs to understand that once a removal procedure begins, the mercury filling will spew enormous quantities of invisible, odorless, poisonous gas. This must be captured in a vacuum and removed from the vicinity of the dental personnel and the patient. Some poorly designed dental offices have vacuums that exhaust indoors, like a closet or utility room. This is wrong, because the whole office then becomes contaminated as a result. The next important step is to protect the breathing zone. This can be done for the doctor and assistant with a Mine Safety Association mask with an additional small particle filter. An even better system is like the one used for hazmat disposal experts or firemen. This provides fresh air under positive pressure. This avoids the principal problem with masks alone, which is termed suck-by, where contaminated air is inhaled around the edge of the mask instead of through the filters. For the patient, it presents a slightly different problem. Positive pressure air is strongly recommended as well. A suitable substitute is a nasal hood, similar to that used to administer nitrous oxide analgesia, but without the positive pressure. This reduces the exposure, but a rapid inhalation can suck by the edge of a nasal hood. Therefore, positive pressure is considered safer. Gloves, nitrile are preferred to latex because mercury does not appear to penetrate nitrile gloves as readily as it does latex. Personally, I prefer to use a rubber dam for many reasons, but some dentists and a few patients prefer packing the mouth with cotton. It's not important because both those procedures are approved, but remember, do not breathe in through your mouth at all, ever, during any dental procedure. Before commencement of the drilling, the patient holds a supplemental vacuum close to their chin, and the assistant should place her high-volume vacuum within 1/2 inch of the tooth. And that's not the saliva ejector, it's a big vacuum. It should remain glued to that side of the tooth until every scrap of mercury filling and the particles have been thoroughly removed and rinsed away. In addition, since mercury easily penetrates latex, a vacuum such as a saliva ejector should be placed underneath the rubber dam, and the patient should be cautioned not to breathe in through their mouth. A technique pioneered and taught by the International Academy of Oral Medicine and Toxicology called the cut and chunk technique involves sectioning the old filling with a burr. In the diagram, you see the burr is placed into the filling and drawn across, cutting the filling into 2 pieces. That reduces the amount of drilling necessary to remove the filling. No diamonds, please, as that creates small particles and is potentially even more dangerous than using a burr. The dentist, using a full water spray from his handpiece and a supplemental spray from the dental assistant with her water syringe, carefully cuts the old filling in half and removes the 2 pieces. This minimizes the drilling necessary, and the water cools the filling to reduce the mercury vapor. After the filling is out, the dam and cotton rolls should be removed and the mouth thoroughly flushed for about a minute in order to remove all the particles.
We're very careful about telling patients that they better get the fillings taken out by somebody who knows how to do it properly, and we make sure that the materials they put in are ones that are biocompatible and safe before they end up exposing themselves. It's very dangerous to have fillings removed by somebody who just has a que sera attitude about putting them in.
When taking out mercury fillings, everyone is exposed to high levels of the poisonous mercury vapor. The dentist, the dental assistant, and you, the patient. When having your amalgams removed, you have a right to insist that your dentist protect you from those mercury vapors.
The major problem with mercury in all of the studies that have ever been done, like in the Minamata Bay disease, of the people that were exposed to toxic fish, the individuals that were affected the most were the babies that were in utero when the mother ate the fish. And that's because mercury concentrates in the fetus. A recent study from Sweden has shown that mercury in the cord blood that feeds the infant is double the mercury level of the mercury in the mother's blood. That's indicating that the cord blood is concentrating the mercury from the mother and is feeding the fetus, and it gets into the fetus's body. Now, our studies on the mercury in the birth hair of autistic versus controlled children, with regard to the controlled children, the level of mercury in their birth hair went up with the increased number of birth mothers' amalgam fillings. So there's almost no doubt that mercury in the birth mother, in a pregnant mother, concentrates in the fetus and can cause damage to that fetus. Mercury is neurotoxic. It has no business being in our food, and it certainly has no business being in our dental and medical treatments.
How mercury causes brain neuron degeneration. Mercury has long been known to be a potent neurotoxic substance, whether it is inhaled or consumed in the diet as a food contaminant. Over the past 15 years, medical research laboratories have established that dental amalgam tooth fillings are a major contributor to mercury body burden. In 1997, a team of research scientists demonstrated that mercury vapor inhalation by animals produced a molecular lesion in brain protein metabolism, which was similar to a lesion seen in 80% of Alzheimer's diseased brains. Recently completed experiments by scientists at the University of Calgary's Faculty of Medicine now reveal, with direct visual evidence from brain neuron tissue cultures, how mercury ions actually alter the cell membrane structure of developing neurons. To better understand mercury's effect on the brain, let us first illustrate what brain neurons look like and how they grow. In this animation, we see 3 brain neurons growing in a tissue culture, each with a central cell body and numerous neurite processes. At the end of each neurite is a growth cone where structural proteins are assembled to form the cell membrane. 2 principal proteins involved in growth cone function are actin, which is responsible for the pulsating motion seen here, and tubulin, a major structural component of the neurite membrane. During normal cell growth, tubulin molecules link together end to end to form microtubules, which surround neurofibrils, another structural protein component of the neuronal axon. Shown here is the neurite of a live neuron isolated from snail brain tissue, displaying linear growth due to growth cone activity. It is important to note that growth cones in all animal species, ranging from snails to humans, have identical structural and behavioral characteristics and use proteins of virtually identical composition. In this experiment, neurons also isolated from snail brain tissue were grown in culture for several days, after which very low concentrations of mercury were added to the culture medium for 20 minutes. Over the next 30 minutes, the neurite membrane underwent rapid degeneration, leaving behind the denuded neurofibrils seen here. In contrast, other heavy metals added at this same concentration, such as aluminum, lead, cadmium, and manganese, did not produce this effect. To understand how mercury causes this degeneration, let us return to our illustration. As mentioned before, tubulin proteins link together during normal cell growth to form the microtubules which support the neurite structure. When mercury ions are introduced into the culture medium, they infiltrate the cell and bind themselves to newly synthesized tubulin molecules. More specifically, the mercury ions attach themselves to the binding site reserved for guanosine triphosphate, or GTP, on the beta subunit of the affected tubulin molecules. Since bound GTP normally provides the energy which allows tubulin molecules to attach to one another, mercury ions bound to these sites prevent tubulin proteins from linking together. Consequently, the neurite's microtubules begin to disassemble into free tubulin molecules, leaving the neurite stripped of its supporting structure. Ultimately, both the developing neurite and its growth cone collapse, and some denuded neurofibrils form aggregates or tangles, as depicted here. Shown here is a neurite growth cone stained specifically for tubulin and actin, before and after mercury exposure. Note that the mercury has caused disintegration of tubulin microtubule structure. These new findings reveal important visual evidence as to how mercury causes neurodegeneration. More importantly, this study provides the first direct evidence that low-level mercury exposure is indeed a precipitating factor that can initiate this neurodegenerative process within the brain.
We come to a conclusion is that people who suffer from certain neurological diseases such as autism and Alzheimer's disease represent a subset of the population that for some reason, possibly genetic, they are unable to get mercury out of their cells. When the mercury gets into their bloodstream, it soaks into the cells immediately, and a healthy person or a person that can get rid of the mercury will combine that with a material in the normal cell and get rid of it, whereas someone who is unable to excrete mercury will retain that mercury and subsequently becomes afflicted.
Placing a mercury filling in my child to me constitutes an assault on my child. We know mercury comes out of these fillings. Other countries have stopped placing these mercury fillings in their children. The United States has an obligation to their children to also stop placing mercury fillings.
Mercury pollution in the environment is becoming a serious global problem, and dentists are a significant contributor to this problem. Every time an amalgam filling is placed or removed in a dental office, mercury is going down the drain, into the sewage system, into the water supply, into the fish that we eat back again. This can be stopped fairly easily. Then dental separator systems in the dental office can cut off this flow of mercury into the wastewater. When this has been done community-wide, such as in San Francisco, it showed that over 90% of the mercury going into the wastewater treatment plants was coming from dental offices.
This mercury fight, and I didn't consider it a fight when I first started. I had no dog in the fight to speak of, but I did do research on Alzheimer's disease and found that mercury would mimic many of the biochemical aberrancies that we see in Alzheimer's disease. By treating it into rats exposed to mercury vapor, or taking normal brain and treating it with mercury, we could create biochemistry that looked very similar to what we see in Alzheimer's disease. So at the very least, you could say that if mercury weren't causal, at least it would be an exacerbating factor for these diseases. Recent research that's come out has supported my concept that mercury is probably fundamentally involved in Alzheimer's disease. I don't think there's much doubt about that. This is not popular with the American Dental Association and even perhaps our Food and Drug Administration dental branch. And you cannot get support for any studies showing that mercury is involved in causation of any neurological disease. The people who fund American research, and this is easy to check out, just go to the literature, almost all the papers that talk about mercury being toxic, neurotoxic, will be submitted from foreign countries. They do have a child study where they're putting mercury in the mouths of children. I would think that they should be very critical, and they're using blood levels of mercury and urine levels of mercury to decide exposure, etc. And what our research on the autistic children shows that means nothing. I mean, children that can't excrete mercury will have low urine, low blood levels, and it will indicate that they are not exposed when indeed they are the ones that are the most susceptible. And I would suggest to people who are learned, go back and look at the Shishil's study, where they looked at mercury in children, in an island where they primarily ate fish. Look at the Faroe Island studies in the North Sea, where they again looked at children who had primarily a diet of fish and whale. And what you will see is that the children with the lowest mercury levels in their hair and blood in those studies were the ones that were the most adversely affected, whereas the children with the highest level in their hair and blood were the ones that were the least affected. What this means is that the children who are effectively excreting the mercury, are getting better and are not getting sick, whereas the children who can't excrete it are the ones that are having medical problems and neurological problems. Removing amalgams does make the population in general healthy. And when the ADA says there's no correlation between amalgam placement, amalgam existing in your mouth, and poor health, they're just ignoring the literature that they don't want to look at. Because you understand, Alzheimer's disease, autism, ALS, MS, Parkinson's are all major diseases that we haven't a clue about what caused them. We have no absolute proof. And it's not because we can't intellectually figure that out. It's because we are not given the funding to pursue the fact that maybe heavy metal toxicity, predominantly mercury toxicity, is the cause of these illnesses.
These instructions can be found on the webpage of the International Academy of Oral Medicine and Toxicology. I would recommend that you review them and have your dentist do the same before commencing any procedures involving mercury.